We don't care about that. Okay, so on Friday, we were talking about the different reactions of photosynthesis. I did post on Moodle this diagram and the basic one from cellular respiration that are there that are blank. So if you want something quick to print out for practice labeling and thinking about where things are happening and what's kind of the overview of what's happening in the different steps of cellular respiration and photosynthesis, those are both there on Moodle on a blank PowerPoint. So we talked about on Friday, we talked about the light reactions and what's the main product of the light reactions. So thinking about the light reactions, what are we making in light reactions? What are we starting with first? We're starting with water, and what do we get out of that water? We get some oxygen, and what else? Yes, what? What? Hey, what? I think it's ATP. ATP, yes. So we make some ATP. What process, what basic thing are we doing to make ATP that looks similar to cellular respiration? We have what processes make this ATP? We have embedded in the membrane of the thylakoid, what is that? In that membrane of the thylakoid there, what is going on to make ATP? That should remind you a lot of cellular respiration. What's embedded in that membrane? So we have chemiosmosis. So we have that hydrogen ion gradient and the hydrogen ions then passing through the ATP synthase to provide the energy to phosphorylate ADP to make ATP. What do we do to make the hydrogen ion gradient? That also happens in the thylakoid membrane. That also should remind you of cellular respiration. How do we get a hydrogen ion gradient? We're building up a higher concentration, I agree. How do we get that? Because we're making a concentration gradient, we're making a higher concentration of hydrogen ions on one side of the membrane than the other. That's active transport, right? We're moving from low concentration to high concentration. So what process will allow us to pump those ions across the membrane in the wrong direction? The movement of electrons. Yeah, we have an electron transport chain. So we've got this electron transport chain. The electrons are moving down the electron transport chain. That's providing the energy that we need to pump those hydrogen ions across to make a chemiosmosis concentration gradient to be able to generate ATP. And then remember this diagram? Not that one. So this is our first, we add the light, we add the light energy, we boost that electron energy up, and that energy is, those electrons are used to make ATP. And then, in addition to that, we have another photon of light coming in. I'm pointing to this and you can't see that. Another photon of light is coming in to boost that electron up again, boost another electron up. And that electron is going to be used to do what? Where is it going? We made ATP from photosystem 2, and then photosystem 1, what are we going to make? Never mind my electron. Oh, wait. What's in the bucket? NADPH, what is that? Is it full or empty? We're filling electron carriers. So at the end of the light reactions, we haven't even touched carbon dioxide. We have split water, use some electrons to make ATP, and use some more electrons to fill electron carriers. And we're saying 9 because that's what we're going to end up using in the next steps. So I think we had just started the challenge cycle, right? Yes. Okay, good. So we've gone from photosystem 2 to photosystem 1, we filled electron carriers, we made ATP, and now this ATP and these full electron carriers are going into the Calvin cycle. We already talked about chemiosmosis. Where did I put the pen down? Sorry, I'm trying my pen. So this ATP, that is not a pen anymore. This ATP and these NADPHs are going into the Calvin cycle. What is this top molecule of the Calvin cycle? It's the other reactant for photosynthesis. Light plus water plus carbon dioxide. So we're taking carbon dioxide into the Calvin cycle. And this is what the Calvin cycle looks like. And I need to back up a couple of things. I changed some numbers on here because I was getting myself confused. The first phase of this is carbon fixation. And we said that fixating means we're taking it from a gaseous form in the atmosphere and putting it into a useful, usable form of carbon, organic, into an organic molecule. So this is our carbon dioxide. We are going to go through this 3 times. Okay, we have to go through this phase, the cycle 3 times. How many times do we go through the citric acid cycle for each molecule of glucose? 2. So the citric acid cycle is 2. The Calvin cycle is 3. I don't have any great ways for you to remember that one. So we're going to start talking about this. We're going to go back through this again, and we're going to talk about, I think I said this wrong on Friday. This is what I lay in bed on the weekends and think about. I think I said this wrong. So Rubisco is what? Rubisco is the enzyme that's going to catalyze the reaction between carbon dioxide and Rubisco and RUBP. So Rubisco is the enzyme, and we're going to take that carbon dioxide and we're going to add it to RUBP, which is a 5-carbon molecule. And I think I said RUBP was the enzyme and I was wrong. So we're adding carbon. We've got a 1-carbon molecule. We're going to add to a 5-carbon molecule. That's going to give us how many carbons? We have a 6-carbon molecule here. We're not worrying about the name of this. This is just an unstable form. So because it's unstable, that's going to make it split. So this is 1 6-carbon molecule. We're going to split it. So now we're going to have what? We split a 6-carbon molecule in half. We're going to have 2 3-carbon molecules. So that 3-carbon molecule is phosphoglycerate. And you can see it's got a phosphate group on the end. So we have 2 of these. So in the next step, we're going to take off this phosphate group and we're going to add it to, nope, we're not, sorry. We're going to use ATP, sorry. We're going to use ATP. I just want to phosphorylate stuff. We're going to use ATP here and we're going to add another phosphate group to the other end. And because we have 2 of these, we're going to use 2 ATPs. But how many times are we doing this? We're going through the cycle 3 times. So how many ATPs am I really using? 6 ATPs. So now I have 2 3-carbon molecules that have phosphate groups on either end. Now I'm going to use some of my NADPHs, because we're in at least that helps. In photosynthesis, you can remember it's pH in it because it's photosynthesis. It's P photosynthesis for the electron carriers. So we are going to do what if we're taking electrons away from them? What are we doing to them? We're going to oxidize them. So this is an oxidation reaction. We're going to oxidize the NADPHs to make them NADPs. And in that process, we're also removing the phosphate group that we just put on. I can't explain to you molecularly why all this makes sense. Why are we adding a phosphate group and then taking it away right away afterwards? Biochemistry will explain this to you. Okay, so we are taking away 2 NADPHs. But we're going through the cycle 3 times, so we're taking away 6 NADPHs, really? Because we're going through the cycle 3 times. So for each of those 1,3-biphosphoglycerates, we're going to empty 2 electron carriers, and that's going to happen 3 times. So from this, now we have made 6 NADP pluses. So in the Calvin cycle, we are making empty electron carriers. Why is that important? What are we going to do with them again? We're recycling them. We're going to be able to fill them up again in the light reactions. So we're reusing. The cell is efficient. So we're oxidizing those, and we have produced these phosphate groups. This is the reduction phase. NADPH is being oxidized, but this is gaining electrons, so this is being reduced to this. That's why it's called reduction. Now, from this, we have 2 G3P molecules. G3P is glyceraldehyde 3-phosphate. So we've made 2 G3Ps, but we're going through this 3 times, so we've really made 6 of them. So one of those is going to go out, and that's going to be the output from this. That's our carbohydrate that we made. So photosynthesis makes carbohydrates, and we are making 1 G3P molecule. We made 6, but we're keeping 1. The other 5 G3P molecules are over here. So out of these 6, 1 goes here, and 5 of them go here. And this is phase 3 of the Calvin cycle, which is the regeneration of RUBP. We have to make more of it so we can keep going. So we can keep the cycle going. So this is what makes it a cycle. So there's a series of steps during which we take 1 ATP molecule for each turn of the cycle. And how many times are we going around? 3. So here go our other 3 ATPs. So all the ATP that was made is now used. And we have 1 molecule, well, really 3 molecules of RUBP, which can start the cycle all over again. Yes. Regenerations. I think we accidentally made too many electron carriers. Sorry, it's 9 ATPs and 6. We're not using these, so I think that I don't think we needed them. So in your yellow, on your yellow chart, for the Calvin cycle, where is this happening? Where in the cell does this happen? It's happening in the stroma. So that's the area inside the inner membrane, but outside the thylakoids. And what is going into the Calvin cycle? Carbon dioxide, ATP, and NADPH. Okay, so we're putting in carbon dioxide, that's gonna be the carbons that will be the source of carbon for our carbohydrate that we're making. We're putting in ATP, which is gonna provide the energy for those Calvin cycles that happen. And we're putting in full electron carriers, which will provide the electrons that we need for the reaction to happen. We're also adding, you can think about the fact that we're adding RUBP, the 3 of those 5 carbon molecules to start it. At the beginning, we have to have these. So they're kind of coming in here. What do we get out of this? We get NADP plus, so we get some empty electron carriers. What else do we get? We do get some inorganic phosphate groups. What is the point of this reaction? What did we make? We made a carbohydrate. We made G3P. So the point of the reaction is that we made G3P. G3P is then going to go on to become, it will be used, it will be processed, maybe processed is a better word. To become monosaccharides, which then can become disaccharides, which can become polysaccharides. So we made a carbohydrate. We emptied some electron carriers. We made RUBP again because we regenerated at the end. So we start with RUBP and then we also make more RUBP. In terms of carbons, where are our carbons going in this reaction? We're bringing in carbon dioxide. And we're bringing in RUBP. So if you do carbon calculations, we have carbon dioxide and we have RUBP, which is a 5 carbon molecule. And at the end, we end up with G3P, which is a 3 carbon molecule, and another RUBP molecule. Well, 3 of them actually, but. So we didn't lose any carbons, we just spent a lot of time rearranging carbons and getting one carbon that we want as output and then just regenerating the other one so we can keep on going and going and going and going. Am I doing this? No, I'm not doing it right still.
You're facing a giant bowl of energy-packed carbon crunchies. 1 spoonful, 2, 3. Soon, you're powered up by the energy surge that comes from your meal. But how did that energy get into your bowl? Energy exists in the form of sugars made by the plant your cereal came from, like wheat or corn. As you can see, carbon is the chemical backbone, and plants get their fix of it in the form of carbon dioxide, CO2, from the air that we all breathe. But how does a plant's energy factory, housed in the stroma of the chloroplast, turn a 1 carbon gas like CO2 into a 6 carbon solid like glucose? If you're thinking photosynthesis, you're right. But photosynthesis is divided into 2 steps. The first, which stores energy from the sun in the form of adenosine triphosphate, or ATP. And the second, the Calvin cycle, that captures carbon and turns it into sugar. This second phase represents one of nature's most sustainable production lines. And so with that, welcome to the world's most minuscule factory. The starting materials, a mix of CO2 molecules from the air and pre-assembled molecules called ribulose bisphosphate, or RUBP, each containing 5 carbons, The initiator? An industrious enzyme named Rubisco that welds one carbon atom from a CO2 molecule with the RUBP chain to build an initial 6-carbon sequence. That rapidly splits into 2 shorter chains containing 3 carbons each and called phosphoglycerates, or PGAs for short. Enter ATP, and another chemical called nicotinamide adenine dinucleotide phosphate, or just NADPH. ATP, working like a lubricant, delivers energy, while NADPH affixes 1 hydrogen to each of the PGA chains, changing them into molecules called glyceraldehyde 3 phosphates, or G3Ps. Glucose needs 6 carbons to form, made from 2 molecules of G3P, which incidentally have 6 carbons between them. So sugar has just been manufactured, right? Not quite. The Calvin cycle works like a sustainable production line, meaning that those original RUBPs that kicked things off at the start need to be recreated by reusing materials within the cycle now. But each RUBP needs 5 carbons, and manufacturing glucose takes a whole 6. Something doesn't add up. The answer lies in one phenomenal fact. While we've been focusing on this single production line, 5 others have been happening at the same time. With 6 conveyor belts moving in unison, there isn't just 1 carbon that gets soldered to 1 RUBP chain, but 6 carbons soldered to 6 RUBPs. That creates 12 G3P chains instead of just 2, meaning that altogether 36 carbons exist, the precise number needed to manufacture sugar and rebuild those RUBPs. Of the 12 G3Ps pooled together, 2 are siphoned off to form that energy-rich 6-carbon glucose chain, the one fueling you via your breakfast. Success! But back on the manufacturing line, the byproducts of this sugar production are swiftly assembled to recreate those 6 RUBPs. That requires 30 carbons, the exact number contained by the remaining 10 G3Ps. Now, a molecular mix and match occurs. 2 of the G3Ps are welded together, forming a 6-carbon sequence. By adding a third G3P, a 9-carbon chain is built. The first RUBP, made up of 5 carbons, is cast from this, leaving 4 carbons behind. But there's no wastage here. Those are soldered to a 4th G3P molecule, making a 7-carbon chain. Added to a 5th G3P molecule, a 10-carbon chain is created. Enough now to craft 2 more RUBPs. With 3 full RUBPs recreated from 5 of the 10 G3Ps, simply duplicating this process will renew the 6 RUBP chains needed to restart the cycle again. So the Calvin cycle generates the precise number of elements and processes required to keep this biochemical production line turning endlessly. And it's just one of the hundreds of cycles present in nature. Why so many? Because if biological production processes were linear, they wouldn't be nearly as efficient or successful at using energy to manufacture the materials that nature relies upon, like sugar. Cycles create vital feedback loops that repeatedly reuse and rebuild ingredients, crafting as much as possible out of the planet's available resources. Such as that sugar, built using raw sunlight and carbon, converted in plant factories to become the energy that powers you and keeps the cycles revolving in your own life.
So that shows you a little bit more about how the RUBP is regenerated. It's a complicated math process. And I'm not sharing the screen again. Oh, someday I will master Zoom. Okay. Questions about photosynthesis? No. Okay, I want to go back a little bit to talk a little bit about, well, yeah, we're gonna skip that for it. Oh, so let's go back to cellular respiration for a minute, because I want to make sure that we talk about the Calvin cycle in a way that I don't think we did. I think we kind of just went through really quickly and said, oh, look, it makes some ATP and it makes some NAD H's and FAD, H2s, and we're gonna have to worry about the details, but I think we do need to talk about the details a little bit because I want you to see the similarities that it has to the Calvin cycle. So we're gonna go back to this. So quick review of cellular respiration. What's the first step of cellular respiration? We start with glucose and what do? Glycolysis. We're gonna split up the glucose. We're gonna make pyruvates. And we're gonna make a little bit of ATP. What are the two phases of glycolysis? Energy investment phase, where we put in some what? ATP, and the energy payout phase, what are we getting out of that? We're making ATPs and two NADHs. So the energy payoff is we get a little bit of energy and we get some full electron carriers. The pyruvate then is oxidized in the process called pyruvate oxidation. Second step. Sometimes we also call that the intermediate step. What do we make in the pyruvate oxidation phase? We turn pyruvate into, how about we spell it right? Yep, the acetyl-CoA. So that's our 2-carbon molecule that we attach the CoA, which is a cofactor to. What do we get out of that? We get some more electron carriers, which we're not really writing on here, but we are now. And we also produce carbon dioxide. And that's when we move, this acetyl-CoA then moves into the third step, which is the citric acid cycle, the TCA, the Krebs cycle, whichever you like to call it. From that, what are we gonna make? We're gonna make some ATP. Not much, but a little bit. And we're gonna fill some more electron carriers. I think that maybe that's what's supposed to go up here. Yeah. Remember, we're filling both kinds of electron carriers in the citric acid cycle. And all of that's going to the last step, which is oxidative phosphorylation. If you ever want to sound smart, just say that. I feel like I'm undergoing oxidative phosphorylation right now. It might sound like a bad condition, but it's good. Okay, and from that, we're going to get lots of ATP. This also produces some carbon dioxide. So when we talked about the citric acid cycle, we kind of just went through, we had a very generic diagram that we went through. I don't want you to panic when you see this and freak out about this. I just want to make sure that we talk a little bit about what this means in terms of how it's similar to the Calvin cycle, because it is similar, we just didn't talk about it. So this is showing you here's the pyruvate, here's pyruvate oxidation, we're making acetyl-CoA, and this is showing you how acetyl-CoA comes in. Acetyl-CoA has how many carbons? 2. So this is a 2-carbon molecule. That 2-carbon molecule, when we start the citric acid cycle, is going to combine with citrate, which is a 4-carbon molecule. Okay, that should sound familiar from the Calvin cycle. In the Calvin cycle, we have a 1-carbon molecule that we combine with a 5-carbon molecule. Here we're taking a 2-carbon molecule and combining it with a 4-carbon molecule. So each time we're making how many carbons? 6. So we have a 6-carbon molecule. There's a lot of rearranging that's happening in the citric acid cycle. Again, just like it was in the Calvin cycle, we see a lot of things happening. We lose water, we add water. We're going to fill electron carriers. We're going to fill some more electron carriers as we move through. We're filling electron carriers, and we are rearranging and rearranging and rearranging. This is an 8-step process. And we are also going to be releasing some carbon dioxides. So here we're releasing carbon dioxide. And we're rearranging to the points. We're gonna have a 6 carbon molecule here. So this is 6 carbon. We're gonna release a carbon dioxide molecule here. And how many times do we go around? 2 times. So we're gonna release another carbon dioxide molecule. That says CO2, wow. And we're gonna end up regenerating at the end. Okay, now see, I should not talk. Oxaloacetate is the 4 carbon molecule. We regenerate this, we put acetyl-CoA and oxaloacetate together and we get a 6 carbon citrate. Okay, so this is the first step, is citrate synthesis. What I want you to get from this is not, again, we're not gonna memorize all these names like I can't even say it, aconatate, or diisocitrate, or alpha ketoglutarate. You will memorize those in biochemistry. What I want you to understand is that we're starting with the carbon molecule, which we're combining with an existing carbon molecule. We're bringing in a carbon molecule, combining with a carbon molecule, and going through this cycle, which as we rearrange, we're going to fill electron carriers and release carbon dioxide, but regenerate this 4 carbon molecule so that we can keep the cycle going. That's what makes it a cycle. And I don't think we talked about the fact that we're combining the acetyl-CoA with another carbon molecule and then regenerating that, just like we saw in the Calvin cycle. We see it in the citric acid cycle. And I wanted to make sure that we understand that, because I think that we also did not talk enough about this step. Not this step, but this concept. Thinking about the fact that you don't just eat glucose. I hope your diet is not primarily glucose. And we kind of breezed through this too. Your diet is composed of different kinds of molecules, and your body has to be able to access the energy from these different kinds of molecules. People who are on keto diets eat only proteins and fats, primarily proteins and fats. So their body has to be able to access that energy because they're not having an excess of carbohydrates. So we've talked about carbohydrates again with the idea of you're starting with glucose, but you're not starting with glucose all the time. And the sugars that you eat include a lot of polysaccharides and disaccharides. Sucrose is a disaccharide. Fructose is a monosaccharide. But starches, things like that are all going to come in through this carbohydrates. So this is talking about monosaccharides, disaccharides, and polysaccharides. Your body's going to take disaccharides and polysaccharides and break them down into monosaccharides, and they're able to just go through this process. So they're able to enter through glycolysis. When you're eating proteins, you know that proteins are composed of what? Amino acids. So most of the proteins that you eat, your body's going to take those amino acids and it's going to break down the proteins into amino acids and then use those amino acids to make new proteins, the proteins that it wants. So most amino acids are used for making more proteins, but not all of them are. So some of them are converted by enzymes to be able to enter cellular respiration at different stages. So these amino acids, if you remember the structure of an amino acid, what's in the middle? It's got a carbon. On one side, it's got an amino group. What's on the other side? We're going way, way back. Chapter. We've got a carboxyl group. And then we've got the side chains. So in order to be able to put it into this process, we have to take off this amino group. So these enzymes, one of the functions of the enzymes is deamination, not deanimation, but taking off that amino group. So then what we're left with is primarily carbons, hydrogens, and oxygens, which looks a lot like a carbohydrate. and can enter, again, depending on how it's been processed, can enter cellular respiration at the end of glycolysis as it moves from glucose to pyruvate. It can enter in pyruvate oxidation, or they can enter in the citric acid cycle. So again, depending on how they're processed, that will determine where they can enter. But these processed amino acids that have been processed by enzymes are able to enter any of those three phases of cellular respiration so that they can be used for energy as well. Fats, we said, were not true polymers. Fats are made up of two different kinds of molecules, that glycerol backbone and then the fatty acid tails coming off it. So they're broken down into those two building blocks, the glycerol and the fatty acids. Fatty acids, glycerol is, you can just phosphorylate it and put it right into the intermediate, what are the intermediate steps of glycolysis? So the fatty acids can enter right into glycolysis also. Fats have more energy than carbohydrates. They have more calories than carbohydrates and proteins. So they're high energy molecules. So it's useful to be able to use them for energy. The fatty acids are actually oxidized through a process called beta oxidation. And this turns them into two carbon molecules, which can be used like the acetyl-CoAs. They can be turned into acetyl-CoAs. So the beta oxidation process turns them into two carbon molecules. They're really long hydrocarbon chains. They can be chopped up and you can add some acetyl-CoAs add some CoAs to them. And then you can put them right into the acetyl-CoA pyruvate oxidation step. So your body is not limited to just one source and has ways to process all these different kinds of macromolecules to make them useful for energy. All of these are what kind of process? Catabolic or anabolic? All of these are talking about breaking things down. So all of these are catabolic processes. Some of these raw materials are not used for catabolic processes like energy making. Some of these are also harvested to be or diverted for anabolic processes. So anabolic reactions can use these raw materials to build things like fat storage. Okay, to store molecules as fat, or making the proteins like we talked about. Those are anabolic reactions that these raw materials can be diverted to use for. So they can be used for catabolic reactions and enter into cellular respiration, or they might be diverted to use for anabolic reactions that are helpful for building up the body. And then the last thing that I wanted to talk about, make sure that we talked about a little bit more carefully, because I think we had like 3 minutes when we talked about this, was the feedback mechanism. Feedback mechanisms are a way for the body or the cell to prevent waste. This is an efficiency measure. Okay, so your cell's not going to be making a whole bunch of stuff it doesn't need, because that's a waste of energy and raw materials. So the feedback mechanisms allow the body to say, hey, I need this, so we're going to make it, or I don't need this, so we're going to stop making it. It's a supply and demand mechanism of regulation. And this often happens through what we call feedback inhibition. All that means is that whatever reaction is taking place is that the product of that reaction will stop. Once you've got enough of that product, it'll stop the reaction from happening. So the final product of the reaction will inhibit the reaction from occurring. at some earlier step, usually by inhibiting an enzyme, some kind of enzyme inhibition. So you can see in this diagram, that's what's going on. We start with glucose, we go through glycolysis, and we go through the oxidation of pyruvate, the citric acid cycle, and oxidative phosphorylation. And the products at each of these levels are ATP. When you get enough ATP produced, the ATP itself will actually stop this enzyme from functioning. Phosphofructokinase, or PFK. So phosphofructokinase, you have to say them slowly, or you don't know what you're going to say, is an enzyme in the first 3 steps of glycolysis that is active, and it will be inactivated by the presence of ATP. So if this enzyme's not functioning, nothing else after that's going to work. So if you have enough ATP or too much ATP, you can stop this enzyme from working at any stage of the at any stage of cellular respiration. On the other side, you can also see another molecule that can inhibit phosphofructokinase, I'm just going to say PFK, is citrate. So when we think about the citric acid cycle makes citrate. If there's a lot of citrate in the citric acid cycle, that will actually inhibit this PFK enzyme from working. So there's 2 mechanisms, 2 feedbacks that will say, hey, we don't need to do any more cellular respiration. We can stop where we are. We don't need any more glycolysis. We don't need to continue using the resources of the cell to keep making ATP because we've got enough. So if there's a lot of citrate or a lot of ATP, it's going to inhibit that PFK enzyme in glycolysis. On the other hand, if you're in a situation where you need more ATP because the cell's doing a lot of work, especially in things like muscle cells, there are ways to stimulate it. And that molecule that will stimulate the process of this is AMP. What would you guess that might stand for? We know ATP is adenosine triphosphate, and ADP is adenosine diphosphate. So this is adenosine monophosphate. So this comes when ADP, we have ATP, and we remove a phosphate group, we get ADP. If we remove another phosphate group, we get AMP. So if there's a lot of AMP present in the cell, that stimulates this PFK enzyme to say, hey, we need to make more ATP because we've got too much AMP, which isn't useful for us for doing work. We need more. So the presence of AMP or cyclic AMP will actually stimulate the citric acid cycle. I mean, not the citric acid, sorry, the glycolysis to happen more. So this is the way that the cell can say, I need more energy or I don't need more energy, and can regulate the process to be efficient. So I wanted to make sure we talked about that. We have 3 minutes. I have a couple questions. Review questions. I didn't know how much time we had. Why don't you take a second and see what you can do here, matching each step with where it happens.
Okay, glycolysis, where is it happening? Cytosol. The light reactions, where are they taking place? The thylakoid membrane, oxidation of pyruvate. You just say the letter even. F, cytosol to the mitochondria, Calvin cycle. Calvin cycle is happening in the stroma. Oxidative phosphorylation. The inner mitochondrial membrane and the citric acid cycle. There's only one left. Hey, in matching, you can always at least guess the last one. Okay, citric acid cycle is happening in the mitochondrial matrix. P680 is essential for the transfer of electrons during which process? P680, not P700, is photosystem 2. Again, you could panic when you see this question, but you're talking about transferring electrons. We've not talked about this ever. Krebs cycle has nothing to do with photosynthesis, and the Calvin cycle has nothing to do with electrons. Okay, I will see you on Wednesday for a test. That one? Make sure you sign the attendance sheet because I got here late.